All right, here we're starting with Chapter 10, Section 2. Still on this idea of antimicrobial treatment inside the body. And here are the objectives that we'll cover with this section. Objectives 5 through 9. We ended Section 1 here, looking at the goals of antimicrobial drugs. And what we want to do is to disrupt different cellular processes. And we want to try to be selectively toxic so that we can kill or inhibit the actions or the synthesis of molecules in microorganisms, but not the host cell. Keep the host healthy. And here is a list, the antimicrobial drug categories of the major areas that antimicrobial drugs target. And you have this simple list here. But if we move to this figure here, you get the picture version. And you have major groups. First, in the upper left-hand corner is the protein synthesis inhibitors. These are going to act at a variety of different areas within the process of protein synthesis. Most, if not all, are going to act on the ribosomes in some fashion. And the reason this is a good target is because ribosomes are different in prokaryotic cells versus eukaryotic cells. The sizes are different. The shape and activity, or the shape, is different. And so that is a fantastic target because then you can be selective for the toxicity. Kill the prokaryotic cells causing the damage. Leave the eukaryotic cells alone. Then on the bottom left-hand corner, you have folic acid synthesis, inhibition. All right, that's going to occur in the cytoplasm. But really what this idea is to block pathways and inhibit metabolism in general. And folic acid is just a major target because there are different enzymes, or there are enzymes in prokaryotic cells that do this, that allow the bacteria to synthesize their own folic acid, whereas in eukaryotic cells, we don't synthesize our own folic acid, but rather we have to take that in through diet, which is actually pretty easy to do. And in the cases of pregnancy, it is recommended that women have extra folic acid just because folic acid is a major component of DNA synthesis. And if there isn't enough folic acid for the mom and for the developing child, there can be problems that arise. Thankfully, that's not very common, though, just because folic acid is in ample supply. All right, then on the right side, you have cell wall inhibitors. Humans do not have cell walls, so that is also a very easy target for antimicrobial drugs. So it can block the synthesis of the cell wall. It can block the repair if necessary. It can block the passage of the cell wall components from the interior of the cell to the exterior, where the cell wall is going to be built. And then there's the cell membrane. And if you disrupt the cell membrane, you cause loss of selective permeability. And so material can go into the cell freely or leave the cell freely. And that's going to cause probably osmosis issues, right? Water pressure. And then finally, there is the nucleic acid synthesis target. All right, whether it's the DNA or the RNA, you could inhibit replication and transcription. You can inhibit the RNA polymerase activity and then disrupt the ability to make RNA, which will then inhibit protein synthesis because you need the mRNA right here in order to run through your ribosomes to create that protein. All right, this is a picture of the cell wall synthesis. I don't know if this one is actually in your textbook, but I really like it. And if you remember, the cell wall is made up of alternating subunits of the NAM and NAG molecules. All right, the kind of pink and maroon color here. And those are just alternating, and it creates the major component of the cell wall. You think about these kind of as bricks. And then the mortar between the bricks, the cement, are going to be these different cross-linkings. And that is going to stabilize the NAM and NAG subunits and make it a very stable cell wall. Now, if or when the cell is going to go through that synthesis process and growing in order to replicate, there has to be an increase in the size of the cell, even though typically when we talk about growth of bacteria, we're talking about replication. But it does have to grow a little bit because it's going to divide. And so you can see the size difference where the parent cell had to add more cell wall to itself so that it gets bigger, and then it's going to replicate or divide. All right, so what happens when the cell is growing, increasing in size? It has to expand that cell wall. And that can be by adding NAM and NAG subunits. And in that process, you also have to add the crosslinks. What some drugs do, like penicillin, it inhibits new cross-linkings from forming. And so as that cell is getting bigger in order to replicate, without the cross-linkings there, you end up with holes in the cell. And then you lose the integrity of the peptidoglycan, and the cell is going to burst from osmotic pressure. And then that cell dies. And the important thing to focus on this idea of using antimicrobials to inhibit bacteria, in this case, on this figure, is that the bacteria cell has to be in that growing process. Otherwise, the drugs aren't going to work, right? If the drug is against the cell wall, the bacteria needs to be building the cell wall in order for it to work. If it's against DNA synthesis, it has to be going through the process of synthesizing DNA in order for that to work. All right, this is one of my favorite pictures. This is a before and after penicillin picture, where you have this little guy here, nice bright white, all right, probably an electron microscopy picture, or maybe dark field microscopy. And it's super white because the integrity of the cell wall is intact, and it's a very solid cell wall. All right, then after penicillin, look at how that has changed. All right, you've got it's not bright white anymore. It almost looks, it's very similar to the background. And all of this dark stuff in here are holes that have been created because the cell has been expanding, but it's lost the integrity of the cell wall. All right, so concept check here. Which of the following antibiotic modes of action will have the least toxic effect on human cells? So antibiotic A is going to act on DNA replication. Antibiotic B acts on the cell membrane. Antibiotic C acts on the peptidoglycan cell wall. All right, so what you should know is that antibiotic A, acting on DNA replication, all right, hopefully that drug is going to be specific for the prokaryotic cells, but our cells do undergo DNA replication. So maybe, just possibly, there might be some interaction. Acts on the cell membrane. Bacteria cells do have a cell membrane. All of our cells are going to have a cell membrane as well. All right, and then antibiotic C acts on the peptidoglycan cell wall. You and I do not have any peptidoglycan. We do not have a cell wall. So even though A and B might be very specific for the prokaryotic activity, we do know that antibiotic C would actually be the best choice because we simply do not have peptidoglycan in our cells. So C would be the answer. All right, then there's this idea of broad spectrum drugs versus narrow spectrum. All right, broad spectrum is simply going to be effective against more than one group of bacteria. Narrow spectrum drugs are going to target a very specific group. And one of those drugs was polymyxin, or one of those drugs is polymyxin. And penicillin is one that was originally narrow spectrum. All right, but it has been altered over the years for the many different generations of penicillins that are out there. So it's not quite as specific as it used to be, or not as narrow spectrum as it used to be, but it's still fairly narrow. It's just actually not used very often. Alright, so here you have table 10.5, and I don't want you to memorize these drugs, but rather understand the idea, the concept of narrow spectrum versus broad spectrum. Alright, the longer the line is, the more groups the drug is going to target. So tetracyclines, this is the hallmark of the broad spectrum antibiotics. It covers rickettsia, chlamydia, gram-positive bacteria, gram-negative bacteria. The only guys that it doesn't cover are tuberculosis, the organism that causes tuberculosis, and a couple of guys that fall outside of these major groups of organisms. Penicillin, gram-positive in general, but it does cross over into a couple of other groups. Polymyxin, very specific. Isoniazid, very specific. Tobramycin, fairly specific. And so the shorter than the line is, the more narrow spectrum the drug is. All right, do you want to take, if you were sick, would you want to take a broad spectrum antibiotic or a narrow spectrum antibiotic? All right, if you knew what you were sick with, all right, you would want to take as narrow spectrum drug as possible and target the infectious organism and get well. All right, the problem with broad spectrum antibiotics is that it's going to kill off not only the organism that is making you sick, but also all of the good guys that are living in and on us. And that can cause problems by killing off our normal flora, because we do have, say, yeast organisms that are part of our normal flora. Yeast is fungus. It's not going to be killed off by the microorganisms or by that antibiotic that is specific for bacteria. And so then the fungal organism can expand in its location because the bacteria are leaving, which leaves more space, more food for that yeast, and it grows up out of normal numbers. And so then a person might develop a yeast infection. And this is fairly common, or most common in females, generally being a vaginal yeast infection. All right, but let's say that you were actually very sick, like in the hospital type very sick, and the doctors really weren't sure what was causing your illness. At that point, a broad spectrum antibiotic is actually best because it's going to cover a lot of areas while the clinical lab is testing your samples, whether it be a urine sample, a sputum sample, a blood sample, to look for the actual organism that is causing the problem. And then at the same time, they're going to look for susceptibility tests to see what drug that organism is causing, or that what drug the organism that's causing the problem is susceptible to. And that's the disc diffusion, or the Kirby-Bauer test that we talked about in section 1. Alright, so this is where we're going to stop for section 2, part 1, and we'll start here with section 2, part 2 of the audio component.